DNA methylation is not involved in. You try answering this question and then I'll explain you this. Yeah, DNA methylation is not involved in chromatin remodeling, RNA splicing, gene silencing, base excision repair. It's not involved in. Is it involved in chromatin remodeling, splicing? Do you know that chromatin is of two types? Euchromatin and heterochromatin. Yeah, chromatin is of two types, euchromatin and heterochromatin. What is euchromatin? It is transcriptionally active region of a chromosome. Do you understand this? It's a transcriptionally active region of a chromosome. When it is transcriptionally active, how will it be? Will it be highly condensed or uncondensed? Yes, it will be uncondensed. I have explained this in detail in the app in regulation of gene expression as well as in chromatin classification. So it's an active region of chromosome and it's highly uncondensed. And because what is the reason behind it being uncondensed? Only when it is uncondensed that can be unwinding, and only after unwinding that can be transcription. So euchromatin is transcriptionally active region of a chromosome, and for it to be active it has to be uncondensed. Whereas heterochromatin is transcriptionally inactive region of a chromosome and it will be highly condensed. When it is uncondensed, euchromatin will stain less densely, heterochromatin stains more densely. That is why whenever you do G banding of your chromosome, G banding or GMSA staining of your chromosome, you find alternative dark and light bands. If you see dark band, it means it is condensed, it means it is inactive, and vice versa. Now, how do you determine whether a region is going to be condensed or uncondensed? There are many mechanisms by which condensation and uncondensation transition is mediated. One is methylation of cytidine residues. Methylation of cytidine residues in CG-rich sequences. So in your genome, there are few regions which are rich in CG sequences. Those regions, if they undergo methylation, it's a sign that it has to be condensed. And when it is condensed, that region is silenced. That region becomes a heterochromatin. So it is a form of epigenetics. Methylation of cytidine residue is a form of epigenetics. What is epigenetic? Environmental effect. Instead of just being dependent on the nucleotide sequences, some changes happen in the genome based on the environmental exposure. That's called as epigenetics. So as you grow, as we evolve, some of the segments of your chromosome undergo methylation of cytidine residues in CG-rich regions. And if a region undergoes methylation, it means it has to be condensed. It means that region has to be silenced. Okay, so methylation of cytidine residues is not involved in. We know for sure it is involved in chromatin remodeling. We know for sure it's a signal for gene silencing. Okay, it is not involved in RNA splicing. Splicing has got nothing to do with DNA methylation. If splicing happens in a segment of chromosome, in an mRNA, it means it's active. Okay, so it's not going to be related to RNA splicing. It is not related to base excision repair. Why? What gives me the confidence to say that it is not involved in base excision repair? What is base excision repair? Suppose this is the backbone of your DNA, okay, and this backbone is attached to nitrogenous bases A, T, G, C, okay. So, and what is between these? You have hydrogen bonds, and this on these nitrogenous bases get attached to a sugar here. Yeah, all these are sugar groups. And I've already told you, the last class I told you the linkage here. What is the linkage here? It's called as beta N glycosidic linkage. This has been this has been explained in detail in the app while I've discussed about DNA structure and about nucleotide chemistry. So what is the linkage which links the base with the sugar? It is beta N glycosidic linkage. And this beta N glycosidic linkage is very weak. So the most common error that happens in a DNA is base excision error, which means this glycosidic linkage is broken and these bases get lost. Yeah, these bases sometimes disappear. In that case, how do you make a repair of it? It is done by glycosylases. Yeah, it is done by glycosylases, which clearly breaks this linkage and removes whatever is left here. And then apurinic or aprimidinic endonuclease. Yeah, apurinic or aprimidinic endonuclease. As the name indicates, it's an endonuclease which makes a nick close to the site where a base has been lost. Yeah, suppose this is where a base has been lost. Apurinic or aprimidinic endonuclease makes a nick here. So that exposes an end. Once an end is exposed, DNA polymerase. Yeah, DNA polymerase, epsilon or beta, they have a proofreading and repair activity. They have exonuclease activity, using which this defective piece of strand will be removed. And the same DNA polymerase epsilon or beta also has got polymerase activity, using which this place or this gap is filled. And then ligase unites the ends. Okay, so I've told you few facts about base excision repair. The most common error that happens in a DNA is base excision error. Reason is the beta N glycosidic linkage which links the base with the sugar is very weak. So many times that linkage gets broken and the nitrogenous base is removed. And the repair is done by apurinic or aprimidinic endonuclease, which makes a nick close to the site where a base has been lost, and that exposes an end. Then DNA polymerase epsilon or beta utilizes its exonuclease activity to remove the defective strand, and the same DNA polymerase utilizes its polymerase activity to substitute a new strand, and ligase will unite the ends. So do we have anything to do with methylase here? No. So methylation is not related to base excision repair. But remember, methylation is related to one repair mechanism, which is mismatch repair mechanism. It is not related to base excision repair, but it is related to mismatch repair mechanism. What is mismatch repair? What is mismatch error? Again, let's consider that these are the two backbones. When I say backbones, you know they are made up of sugar, phosphate alternatively on either side. Okay, and we know the sugar is attached to nitrogenous base. Say A. Here let's have G, T, C. Okay, if you have A, here you expect T, here you expect C. Instead, suppose you find A. Here you expect A, right? Yeah. Here you expect G. So what is mismatch error? What I have shown here, this is a mismatch error. Instead of you finding C, you have found an A. So this is a mismatch error, and how is it repaired? This is repaired again by an endonuclease called as GATC endonuclease. But when an endonuclease, I'll tell you about this endonuclease in a short while, but this endonuclease comes to this place. But this endonuclease will not know which is wrong here. It finds G to be attached to A. It can be either a defect of this A or it can be a defect of G, right? So this enzyme will not be able to find out where the defect is. And how does it identify the defective strand is, the unmethylated strand is considered always as defective. Because based on epigenetic modification of DNA, I am sure the template strand would have undergone methylation randomly. So if a strand is methylated, it means it's an old strand. It means it should have acted as the template strand. And this is the new strand that has been synthesized with a defect. Do you understand this? So methylation is not involved in which repair mechanism? It is not involved in base excision repair mechanism, but methylation is involved in mismatch repair mechanism, wherein the methylated strand is always considered as the template strand, as the normal strand, and the unmethylated strand is considered as the defective strand. So that GATC endonuclease or GATC endonuclease, when it tries to repair the mechanism, it scans sequences on either side of the defective site, and it cuts a point where it finds the nearest GATC sequence, and that exposes an end. And from here, you know, once an end is exposed, DNA polymerase epsilon or beta will start removing this defective strand with the help of its exonuclease activity. And the same DNA polymerase utilizes its polymerase activity to fill the gap, and ligase unites the ends. Okay, so methylation is not related to base excision repair, but it's related to mismatch repair mechanism. And a defect of this mismatch repair mechanism is found to cause hereditary non-polyposis colon cancer. Yeah, it is found to cause hereditary non-polyposis colon cancer. Is it clear now? So DNA methylation is not involved in, it is not involved in RNA splicing, and it's not involved in base excision repair. So it is 2 and 4. Yeah, answer is B. Oxygen utilized is 250 ml per minute. Carbon dioxide produced is 200 ml per minute. What is the respiratory quotient? I would say this is the simplest question that can be asked in INI-CET. Okay, because what is respiratory quotient? Respiratory quotient is equal to carbon dioxide. Respiratory quotient is equal to carbon dioxide produced per minute by oxygen consumed per minute. So what are we trying to check using this formula? So I strongly suggest that whenever you come across a formula, don't start memorizing it immediately. Try to decipher the formula, try to find out what the formula actually means. Try to find out what are we trying to do using this formula. So what are we trying to find out here? Using this, we are trying to find out for this much of oxygen consumed by a body, how much of carbon dioxide is being produced by the body. Do you understand this? What are we trying to check? We are trying to check for this much of oxygen consumed by a person, how much of carbon dioxide is produced by that person. Or in other words, we are trying to check the rate of internal respiration. We are trying to check the rate of internal metabolism. Okay, and that is 0.8 always. In the sense, generally it is 0.8. Reason is oxygen consumed per minute is 250 ml. This is a physiology question that has been repeatedly asked. They ask you this question as, what is the basal oxygen requirement? What is the question? What is the basal oxygen requirement of a person? All of you should be able to answer it any time this is being asked to you. You should say it's 250 ml per minute. Why should you know this fact? Anybody? Why should you know this fact? To set up a ventilator, right? When you set up a ventilator, you should see to it that your ventilator provides this basal oxygen requirement. It should be able to provide at least 250 ml of oxygen per minute. So this is a fact which every one of you should know. Okay, and carbon dioxide produced per minute is 200 ml. So respiratory quotient is equal to 200 by 250, which is 0.8. Okay, But this respiratory quotient would differ depending upon what fuel a person takes. Yeah, if a person is on a carbohydrate rich meal or exclusively carbohydrate meal, what happens when it is digested? What happens when it is metabolized? So glucose C6H12O6 reacts with 6 molecules of oxygen to give rise to 6 carbon dioxide and 6 water. This is a balanced equation which is right. This is what is happening when you take a carbohydrate rich meal. Yeah, glucose gets metabolized by oxygen to give rise to carbon dioxide and water. And what is respiratory quotient? I told you, I said it is carbon dioxide produced permanent by oxygen consumed. Carbon dioxide produced is 6, oxygen is 6. 6 by 6 will be 1. So tell me, what is the respiratory quotient? Yeah, what is the respiratory quotient when a person is on a carbohydrate rich meal? It is 1, and as you were rightly mentioned, respiratory quotient is increased. More carbon dioxide is produced by your body if you are on a carbohydrate rich meal. Okay. Now what happens in a fatty meal full of fatty acids? Palmitic acid C6H32O2. Palmitic acid is the most common fatty acid, right? And the molecular formula for palmitic acid is C16H32O2. This reacts with 23 molecules of oxygen, yeah, to form 16 molecules of carbon dioxide and 16 water. It's always carbon dioxide and water which is produced by complete oxidation of any fuel in your body. So the right side, you can close your eyes and say it's going to be carbon dioxide and water. Okay, so C6H32O2 plus 23 molecules of oxygen would give rise to 16 carbon dioxide and 16 water. Now tell me what happens to the respiratory quotient. How many moles of carbon dioxide are produced? 16. Oxygen is 23. 16 by 23 is 0.7. Respiratory quotient is 0.7 if it's a fatty meal. Yes, it is reduced. Carbon dioxide produced is reduced if you are on a fatty meal. Now protein rich meal, will I be able to give you an equation for this if you are on a protein rich meal? I can't. Why? Because a protein rich meal is made up of multiples of amino acids, right? So I can't be writing a formula for every amino acid and telling you what happens. But I want you to remember that if it's a protein rich meal, the respiratory quotient is 0.8. Yeah, please memorize these numbers. Respiratory quotient in a person who's on a mixed balanced diet. Yeah, respiratory quotient in a person who is on a mixed balanced diet would be between 0.8 and 0.85. Okay, in a carbohydrate rich meal, it becomes 1. Fatty meal, it's 0.7. Protein meal, it's 0.8. So, which has got the highest respiratory quotient? Which has got the highest respiratory quotient? Why? Why do you say it's cholesterol? Oh, okay, yeah, very good. Carbohydrates. So, carbohydrates have the highest respiratory quotient. And there was one question in one AIMS paper, which organ exhibits the highest respiratory quotient? Which organ? For which organ is the respiratory quotient highest? Clue is carbohydrates have the highest respiratory quotient. So, any organ which is dependent on carbohydrates will exhibit highest respiratory quotient, right? And that is brain. You know, for brain, what is the preferred fuel? It's glucose. So, neurons exhibit the highest respiratory quotient. So, what are the facts I've told you related to respiratory quotient? I told you the formula, it's carbon dioxide produced permanent by oxygen consumed permanent. We are trying to find the rate of internal respiration. We are trying to find for this much of oxygen that is consumed, how much of carbon dioxide is being produced by the body. And basal oxygen requirement is 250 ml per minute. Carbon dioxide produced per minute is 200 ml. So respiratory quotient is 200 by 250, which is 0.8. And respiratory quotient will differ depending upon your diet. If it's a carbohydrate rich meal, because it's 6 carbon dioxide that is produced after using 6 oxygen, it becomes 1. Respiratory quotient becomes 1, and it's the highest for carbohydrates. Fat, it is 16 by 23, so it is 0.7. And for proteins, it's 0.8. And the organ with the highest respiratory quotient would be brain. Okay, so what's the answer here? Yeah, 0.8. 1 is for carbohydrates, right? 0.7 is for fat. 1.25, I think they've given you this number 1.25 because they thought you will get confused what should be in the numerator and what should be in the denominator. You try dividing 250 by 200, you will get it as 1.25. Okay, this is just to confuse you. Which of the following is the correct order of steps of PCR? Can anybody tell me? It's a very simple question. The latest question. PCR. Say why she chose A. Denaturation, annealing, elongation, till that she's right. And then why did you choose hybridization? So what are the steps of PCR? I want you to know that PCR is nothing but in vitro replication. Right? It is in vitro replication because it's happening in a test tube. And it's in vitro replication of a desired fragment of DNA. So again in the app I've told you in detail about steps of PCR and types of PCR, about differences of types of PCR. For now it's only about steps of PCR, for which I want you to know that it's in vitro replication. And always for replication the first step would be unwinding. Right? Unwinding is otherwise called as denaturation. So naturally the first step of PCR has to be denaturation, wherein the template strand undergoes unwinding. Suppose this is the template DNA that you have added. So I was telling you that PCR is an in vitro replication process, and for every replication process the first step has to be unwinding, and the other name for unwinding would be denaturation. And suppose the black strand here represents the template DNA that you have added, and let's assume that the red fragment is what you want to be amplified. It's a desired fragment that you're trying to amplify. So the first step is denaturation, which is carried out at a high temperature of 94 or 95 degree, which breaks hydrogen bonds. And after denaturation, you know, in the next step you have to add primers, basically because the DNA polymerase which amplifies needs a primer to be already present for it to elongate the strand. So in the next step you are going to add primers, and these primers anneal to flanking sequences. Yeah, these primers anneal to flanking sequences of both the strands. So the second step is called as an annealing step. Yeah, it is called as annealing step. And I've told you that annealing step temperature has to be low, otherwise the primers won't go and anneal. But at the same time it cannot be too low. If it's too low, primers go and non-specifically bind somewhere else. So annealing temperature has to be optimal. And when I say optimal, it's not a random choice. You cannot just like that say, okay, I'll fix it as 48 to 53. I'll fix it as 53 to 58. So it's not a random number which they choose. Annealing temperature is chosen as 5 degrees less than the melting temperature of the primers. Yeah, annealing temperature is chosen as 5 degrees less than the melting temperature of your primers. Because what if the annealing temperature is higher than the melting temperature of primers? They will melt, they will not anneal. What if it's very less than the melting temperature of primer? They'll non-specifically bind. So optimal temperature for annealing step is melting temperature of primers minus 5 degree. It is you who have chosen these primers. So when you choose primers, when you manufacture it, the manufacturer would provide you the primer. It is provided in a small vial. So when they provide you the primer, they will give you a data sheet for the primer, and that data sheet will include the melting temperature of those primers. Yeah, suppose melting temperature of your primers is specified by the manufacturer, that is 60 degree. What will be the annealing temperature that you choose? 60 minus 5 will be 55 degree, just for an example. Okay. After annealing, in the next step, your DNA polymerase is going to elongate new strands. Yeah, it's going to elongate new strands. So what is the next step of PCR? The next step of PCR is elongation or extension. And the temperature that is necessary for elongation or extension is 72 degree. Yeah, for a conventional PCR technique, it has to be 72 degree. So memorize this. This has been repeatedly asked. It's a fixed question. It's a very simple question to answer. So what are the 3 steps of every cycle of PCR? Every cycle of PCR should go through denaturation step at what temperature? 94 or 95 degree. Annealing at what temperature? 5 degrees less than the melting temperature of primers. Elongation or extension at 72 degree. An annealing step can be otherwise called as hybridization step. Yeah, why do I call it as a hybridization step? Because in this step, the primer hybridizes with the flanking sequence. Okay, so it's not a separate step. So what will be your answer? Denaturation, annealing and extension. Sometimes they give you such fancy or unheard of terms to confuse you, but you have to be very clear. Okay.